and its function, isn't it? Okay. Then you will, there is a question, a written question comes frequently. Briefly mention about the lipid soluble and water soluble substance passing through a membrane. Lipid soluble substance, like a lipid soluble substance, crosses the cell membranes because they can dissolve in the hydrophobic lipid bilayer. For example, oxygen, carbon dioxide, and steroid hormones, etc., are crosses the cell membranes. So they are lipid soluble substance. What are the lipid soluble substance? Say it. What are the lipid soluble substance? I have given an example. Some of the examples: oxygen, carbon dioxide, steroid hormones. Remember, oxygen, carbon dioxide, and steroid hormones. Okay,
Yes, sir.
because they can dissolve in the hydrophobic lipid bilayer. Though it is hydrophobic, but they can dissolve in lipid bilayer. Okay. Water soluble substance, like what are the water soluble substance? Our electrolytes like sodium, chloride, glucose, and water itself is water soluble substance. So water soluble substance, they cannot dissolve in the lipid of the membrane, but may cross through the filled channels or pores, or may be transported by the carriers. There are some carriers like a sodium channel. In the sodium channel, we have sodium potassium carrier. We have glucose transporter. So all those channels and carriers, they take them inside of the cell, though they can't pass through the lipid bilayer. They have a channel. They like you have Howrah bridge like this. You have bridges to cross the rivers. So all those water soluble substances, who cannot pass the membranes, they have those channels, those transporters, those exchanges. Some of the exchanges, like sodium gets out and potassium gets in. Sodium gets in, potassium gets out. So the sodium potassium channel creates. This membranes, we'll get it in details in next, inshallah. You will be asked in the viva, how are lipid soluble substance transported across the cell membrane? You know, diffusion mainly. So by diffusion through the lipid bilayer of the cell membrane. Then you may be asked, how are water soluble substances transported across the cell membrane? This is, I have given some of the answers. Do you have any idea?
Through osmosis.
Uh, in osmosis, what occurs mainly? What in details occurs? By simple diffusion, that is occurs. Facilitated diffusion may occur, and osmosis is the active transport system. The active transport through the carrier proteins also occurs. We'll know in details about the active transport and facilitated active transport and diffusion also in details next, because there is a membrane potential produces in osmosis also. Um, when you have a deep medium and when you have a sluggish medium, they crosses each other. So that is the osmosis. But we will discuss about active transport, facilitated diffusion, and simple diffusion all in the next, in this chapter. But the mechanism of transport of water soluble substance across the cell membrane is by by diffusion and active transport. But diffusion have two parts: simple diffusion and facilitated diffusion. Simple diffusion through the proteins channel, and facilitated diffusion through the carrier proteins. As I said, as the carrier proteins, as I said, water soluble substance are passes through the carriers. So facilitated diffusion occurs through the carrier proteins. Simple diffusion occurs through the protein channels. And active transport through the carrier proteins. Again. So what are those? Can you say one? Anyone, please? Simple diffusion occurs
Diffusion,
through
diffusion and active transport.
diffusion. Active transport is water soluble substance crosses, but simple diffusion through the which protein channel. Facilitated diffusion through carrier protein. Active transport through carrier proteins also. Okay, so by carrier proteins there is facilitated diffusion, active transport. By protein channels there is simple diffusion. Okay,
Yes, sir.
understand?
Yes, sir.
Yes,
Okay.
sir.
Yes,
Yes,
sir.
sir.
And you may be asked in the Viva, how does lipid barrier of the, what is homeostasis?
Sir,
Maintain
homeostasis is the
nearly
maintenance of
constant
nearly constant condition in the
maintenance of nearly
Yes,
constant
exactly. Very
internal
good.
environment. Thank you, sir.
Yes. Okay. So plasma membrane. So you have to have a very clear concept about the homeostasis. When you go to home, you feel safe, and all our body systems tries to maintain the homeostasis. So plasma membrane, how it maintains homeostasis. Plasma membrane maintains cellular homeostasis. It means it is not controls the body homeostasis. It maintains the cellular homeostasis as it covers the cell. So plasma membrane maintains the cellular homeostasis by following way as protective function. As it first protects the cell, so cell damage doesn't occur. Then selective permeability. It has selective permeability. It doesn't allow all those things to come inside the cell or inner the inside the cell. So or in the ICF. So allowing some substance to pass through it and excluding others. That means which are not necessary. They will he will expel it. Okay. So number 3 point is absorption of the oxygen and nutrients and removal of the carbon dioxide and other waste products. So what? Absorption of the oxygen and nutrients and removal of the carbon dioxide and other waste products. By those things, by those three things, you can remember by PSA. P for protective function, S for selective permeability, A for absorption of oxygen, nutrients, and removal of the carbon dioxide and waste products. So by those three things, the cell membranes maintains the cellular homeostasis. Okay. Now we'll go to the what are the functions of membrane protein? It's frequently asked. This is the must question you will have in the written, and it may be asked in the Viva also. This is very important question. Function of integral proteins. So function physiological importance of the cell membrane proteins. It may be asked in as a form of physiological importance of the cell membrane proteins or function of the cell membrane proteins. So when you are asked about this, you will say both of the proteins like we have integral proteins and peripheral proteins. So we'll divide the functions also like this. For functions of the integral proteins, you will say it acts as a channel or pores. Do you know? Do you have recognized? Have you recognized which one is integral or peripheral protein?
Yes, sir.
Yes,
The one
sir.
which is the one which is buried totally, completely in the membrane.
Yes, completely crosses the barrier or membrane. This is called integral proteins, and which are
Yes, sir.
attaches with the proteins or attaches in the side by side. This is called peripheral proteins. So
Arranged in the circular
both
layer.
have different functions. So when you are asked about the functions of the cell membrane proteins, you will say by different with the divisions, like functions of the integral protein first. You will say act as a channels or pores through which water molecules and water soluble substances, especially ions, can diffuse between the extracellular and intercellular fluids. Extracellular remains outside the cell, and intercellular remains inside the cell. So the ions can diffuse between the extracellular and intercellular fluids through the pores in the integral proteins, and again act as a carrier protein. Integral proteins also act as a carrier protein, which transport substances in the direction opposite to their electrochemical gradients for diffusion, and they act as enzymes also for catalyzing reactions at the surfaces of the membrane. At the surfaces of the membrane, act as a receptors of water soluble chemicals such as peptide hormones that do not easily penetrate the cell membrane. Peptide hormones do not easily penetrate the cell membrane, so act as a receptor. Who act as a receptor? Who?
Integral protein.
Tap.
Integral protein.
Protein.
Yes, we're saying about integral protein. So another function is act as a ligands. Act as a ligands that binds to the receptor, causes conformational changes in the receptor protein. In the receptor protein, as we know, it can act as a receptor protein also. So act as a ligand also. How they act as a ligand? They binds to the receptor, causes conformational changes in the receptor protein. Conformational changes, that means that change is its formation also. So that it can act to maintain our body transport system, or our homeostasis mainly of the cell. So 5 points, we have 5 functions: act as a channels or pores through which the water molecules and water soluble substances, especially ions can diffuse between the extracellular and intercellular fluids. And we have they act as a carrier proteins also. They transport substances, act as a enzymes, act as receptors, and act as a ligands. So all these are functions of integral protein. And what are the function of peripheral proteins? Only one. You'll say act as enzymes or as controllers of the transport substance. They controls the transport substance. They are boss. Peripheral protein. So peripheral protein, they bosses. They controls the substances. Okay. So if you are um, boss, if you have to monitor from the upper side. So you're in the chair and you're monitoring. Peripheral proteins are like this, but the manager, that managing director, is integral protein. Okay. So act as an enzyme. They act as an enzyme or act as a controller of transport substances through the membrane, through the cell membrane. You may be asked in the Bible, what are the characteristics of the protein channels? What are the characteristics of the protein channels? Selectively permeable to the certain substances. Selectively permeable. What do you mean by selectively permeable? Selectively permeable. Anyone? What do you mean by selectively permeable? Do you know what do you mean by selectively permeable?
Everything cannot pass.
Not allowed to pass through the membrane. Something is allowed or some are not allowed.
Exactly. Selectively allowed. Then do not allow all those things. So selectively permeable to the certain substances. Many of the channels can be opened or closed by the gates that are regulated by the electrical signals or chemicals that binds to the channel proteins. So characteristics of the protein channel, if you ask about the characteristics of the protein channel, you will say they are, sir or ma'am, these protein channels are selectively permeable. They are not, they don't allowing all those all substances. Certain substances are allowed here to get inside the cell, and many of the channels can be opened or closed by the gates that are regulated by the electrical signals. Some electrical signal comes when they opens, and electrical signal gone, then they are closed. Chemicals that binds to the or chemicals that binds to the channel proteins also. So that are that's all for the characteristics of the protein channel. Cell membrane protein, and write down the functions of cell membrane carbohydrate. Do you know what is the function of the cell membrane carbohydrate? Anyone?
Some of the glycolipids are attached to lipids. Some forms the glycolipids.
Yes, glycocalyx of the carbohydrates are already there. They are named by glycocalyx mainly. So whenever they are attaching with the lipid, they are glycolipid, and when they are attaching with the protein, it is called glycoprotein. So both are glycocalyx mainly, but it is the name, not the function. Function is mainly attachments is the function. Let's
Sir,
see what
receptor for hormone.
what our book says. Most of the membrane carbohydrate have a negative electrical charge. Most of the membrane carbohydrate, the carbohydrate which are attached with the membrane, they have negative electrical charge. So which gives the most cells an overall negative surface charge. So cells have negative surface charge. Always remember, cells have negative surface charge. So when a positive charge is coming to the cells, it is going easily inside and attaches with this. And when it is a negative charge, negative electrolytes, it is going reflecting to its body or its barrier. So most of the membrane carbohydrates have a negative electrical charge and maintaining overall negative surface in the cell. The glycocalyx of one cell attaches, that's an attachment as I was saying, that attachment, glycocalyx of one cell attaches the glycocalyx of another cell. So one cell can attach with another by this glycocalyx. Many of the carbohydrates act as a receptor substance. That is, as we know, proteins act as a receptor, ligands, enzymes, or carrier proteins. But carbohydrate also act as a receptor for the binding hormones. So we have some of the hormones which attaches with the receptors and get signals and produces signals so that our cells can react on those. We have some of the hormones which acts in our body. In deficiencies of some of the hormones, we take drugs or medicines to get those receptors so that our medicines can attach with those receptors and act in our body. So that's how our medicine mainly reacts. Do you know how our medicine reacts? We're saying that the glycocalyx, one of the cells attached, glycocalyx of one cell attaches the glycocalyx of another cell, and many of the carbohydrate act as a receptor substance for binding the hormones. As medicine acts also like this, and some carbohydrates enter into the immunoreactions. Immunoreactions, like we have allergies. Some people have faced a severe allergy. So they faces allergy because of the glycocalyx they have in the cells. They react more to the foreign body. As the food materials or other things, it thinks as a foreign body, so it reacts more. In a Viva, you may ask, give the functions of membrane lipid. You will say, act as a barrier. Membrane lipid act as a barrier that impedes the movement of the water and water-soluble substances. They also permits the movements of the lipids and lipid-soluble substance across the cell membrane. Okay. Now, organelles of the cell. As we know, organelles of the cell, membranous and non-membranous, as I said, nucleus, endoplasmic reticulum, mitochondria, the powerhouse of the cell, lysosomes that creates the lysosomes that has enzymes, Golgi apparatus, peroxisomes have oxygen carrier, and non-membranous bound non-membranous are ribosomes, centrosomes, cytoskeletons. There are three varieties of cytoskeletons like microtubules, microfilaments, and MMI. You can say MMI. That means microtubules, microfilaments, and intermediate filaments. What are those? These are the cytoskeletons. What are the cytoskeletons? Microtubules, microfilaments, intermediate filaments. Please say, what are those cytoskeletons? Anyone? Cytoskeletons. What are the cytoskeletons?
Sir, microtubules, microfilaments, and interskeletal filaments.
Intermediate filaments. Okay.
Intermediate filaments.
Okay. Now mention the 4 cell organelles with one function of each. It is the frequently asked written question. Enumerate the functions of any 2 of the cell organelles there may be asked. So you have to learn the functions of this organelles. Nucleus, gene, DNA, and RNA. It is another topic we need to learn, but it is not important. We need to learn about the what is gene and short note about the gene. The gene, a gene is the discrete portion of the DNA and is the basic. Wait. And is the basic unit of the heredity in a living organism that codes a specific protein, specific protein. So it creates the heredity mainly. As we look like our parents, we are look like our grandfather, grandmother. So there's responsible for hereditary genes. Structures of the eukaryotic genes. We have exons, introns, promoter, terminator, silencer, enhancer. All those things we will read in details in biochemistry, genetics. So don't need in this chapter. Types of genes, mainly dominant genes, codominant genes, recessive genes, carrier genes, sex-linked genes, sex-limited genes. All those things will get in the same the plasma membrane. Next slide, please. All those carbohydrates you can see in this picture. This is the Golgi apparatus. Golgi apparatus, we have another organelle. Another organelle. The correction. You all know the functions of Golgi apparatus. Do you know the function of Golgi apparatus? What they does?
Yes.
Packaging and promoting.
Anyone?
Packaging material to deliver intercellular speech or intercellular target.
Which materials?
Proteins.
Packages proteins mainly. Okay. When proteins are synthesized from the or originate from the ribosomes, ribosomes creates protein. So
protein, glycoproteins and glycolipids.
Glycoproteins and glycolipids.
ঠিক, দুধ পড়ে যাচ্ছে। আচ্ছা, so sorry for the interruption. Functions of Golgi apparatus mainly হচ্ছে conjugation of protein with carbohydrate to form glycoprotein. Conjugation of protein with carbohydrate to form glycoprotein, and as you said, packaging of the protein mainly with ribosome. They grabs it, packages into them, and processes so that they can release outside of the cell. So Golgi apparatus is a very important organelle. So conjugation of the protein, it also acts in conjugation of the proteins with carbohydrate to form the, wait a minute, okay, চলো অফ করছিস কি একটা অবস্থা এরকম তো। Okay, back again. Formation of the lysosomes and secretory vesicles and other cytoplasmic components also by the Golgi apparatus. Lysosome. So please show the next slide. That is the function, mainly conjugation of the protein and the carbohydrate to form the glycoprotein and formation of the lysosomes. You will see the lysosomes next. And this is the endoplasmic reticulum. You can see the endoplasmic reticulum here. Endoplasmic reticulum is a network of tubular and flat vesicular structures in the cytoplasm of the cell. The tubules and the vesicles interconnected with one another. So this is the endoplasmic reticulum. It looks like a reticulum, isn't it?
Does not. Yes, sir.
क्या कहते हैं हिंदी में इसको? जालों को क्या कहते हैं? जाल ही कहते हैं ना?
Yes, sir.
कह रहे जो स्पाइडर की जाल होते हैं वो जाल कहते हैं ना?
Yes, sir. Yes, sir.
Yes, this is the reticulum mainly. Endoplasm network of tubular and flat vesicular structures in the, और एक जाल मिलते हैं हमारे बॉडी में जो क्या
Yes, sir.
Yes, sir.
Okay. Today we have a OT, operation theater at night, about 10 or 10:15. So it is about the case of laparoscopic cholecystectomy. Did you heard ever? Do you have any knowledge about laparoscopic cholecystectomy?
No, sir.
No, sir.
Yeah, you will know when you will go through the anatomy part in abdomen. We have liver and we have gallbladder. You know already we have an organ of named gallbladder.
Yes, sir.
When
Yes,
you
sir.
have any complication, inflammation of the gallbladder, this is called cholecystitis. When you have stone in the gallbladder, this is called cholelithiasis. So when the patient faces these problems, the surgeon must suggest this cholecystectomy operation. So in cholecystectomy operation, it is the removal of gallbladder mainly. So removal of the gallbladder occurs not by opening the abdomen, but just doing some of the holes on your abdomen, some of the key points on your abdomen, and it is done by laparoscope. That means camera inside the abdomen. So there is a hole for the camera port, and the surgeon does input a camera inside the abdomen, inside the patient's abdomen, and another hole just at the G-sternum, as we said. Remember the G-sternum in the anatomy class?
Yes, sir.
Sorry, one mitochondria is called one mitochondrion, and plural form is mitochondria. So the inner cavity of the mitochondrion is filled with a matrix. You can see the matrix. Can you see the matrix in the picture?
Yes, sir.
Yes, sir.
Yes, sir.
Okay. So the inner cavity of the mitochondrion is filled with a matrix that contains large quantities of dissolved enzyme. So enzymes are present here that are necessary for the extracting energy from the nutrients. If you need to extract energy from the nutrients, you need a mitochondria. Okay. So
Yes, sir.
in case of replication, it also helps an inheritance of substance, inheritance of the cells. This is also mitochondrial inheritance may occur, and it almost exclusively maternal. Sperm contributes few in any mitochondria to the zygote, and so the mitochondria is in the zygote comes almost entirely from the ovum. So mitochondria, you can say, from the beginning of life, from the beginning of life, the mitochondria was present because we come from sperms, and sperms when connects with the ovum makes a zygote. That is the true form of starting of life. So when the sperm floats to ovum, it starts its journey. It needs energy. This energy is produced by the mitochondria in the sperm. So it is the most important thing to survival of life. Functions of mitochondria. What is the functions of mitochondria? Production of energy mainly. The adenosine triphosphate, that means ATP. What is ADP? What is ADP?
Diphosphate.
Adenosine
Adenosine
diphosphate.
Adenosine diphosphate. And what is ATP? Adenosine
Adenosine triphosphate.
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 Triphosphate. To adenosine, when an adenosine diphosphate is attached with an inorganic phosphate molecules, that is called adenosine triphosphate. You know, you have gone through in the higher secondary level previously in botany, isn't it? In
Yes,
botany
sir.
you got it. Okay. Did you gone do you have gone through with botany?
Yes, sir.
Okay. uh Inheritance the DNA of the mitochondrion plays a role in controlling replication of the mitochondrion itself, and contains enzyme that take part in respiratory chain, oxidative phosphorylation, TCA cycle, beta oxidation. All those things we have gone through in the higher secondary level in the cycles of the cell, we know. And please show the next slide. That's all for the mitochondria. Remember the functions frequently comes in the written, and we have short note about the mitochondria. If it comes as a short note of the mitochondria, you will say the mitochondria is a powerhouse. Then you will explain the structure, and you will give the function. That's all for the short note. And we have mitochondria. It's also a mitochondria in 3D picture. You can see the matrix, crystal, all those things. In crystal we can find the oxysome that contains oxygen. Outer membrane you can see clearly. Please show the next slide, please. Next slide, please. So this is the lysosome. As we are saying, the lysosomes are the vesicular organelles. From where lysosomes comes the formation of the lysosomes. From where the lysosomes formed? Which organelle
Breaking of
creates
Golgi
lysosome?
apparatus.
Yes, Golgi apparatus. As I said just few minutes ago, formation of the lysosome, secretory vesicles, and cytoplasmic components is the function of Golgi apparatus. You can say Golgi or you can say Golgi apparatus. Anything you can pronounce. Lysosomes are the vesicular organelles that are formed by the breaking off from the Golgi apparatus and then dis- dispersing throughout the cytoplasm. What are the functions of the lysosomes? Go to the next slide, please. Next slide, please. Same slide. It is. And yes, keep this slide. Structures, functions, and types of lysosomes. Lysosomes, mainly the lysosome provides an intercellular digestive system. Intercellular digestive systems. We all have digestive system in our body, but cell needs to digest something. Cell needs to absorb proteins. Absorb the food products or the nutrients to absorb and expel out all those unwanted substances. So this is the digestion process of lysosomes. Uh, Digestion process of the cells. So lysosomes helps to provide the intercellular digestive systems that allows the cells to digest. Damaged cellular structures, food particles that have been ingested by the cells, and unwanted matter such as bacteria is also expelled out by the lysosomes. In viva emacs, the name lysosomal enzymes and their substrates. Some lysosomal enzymes are listed in the table below, like um, ribonuclease, deoxyribonuclease, that means DNA. Um, Phosphatase, those are not DNA. The deoxyribonuclease are the enzymes, and substrate is in DNA. Ribonuclease is the enzymes, the substrate is RNA, and glycosidase enzymes and substrate is complex carbohydrates, glycosides, and polysaccharides. And we have collagenase enzyme, that is the substrate is protein, and we have cathepsins, that is also there is also substrate of proteins. So lysosomal enzymes and their substrates, you may be asked in the viva when you so please respond with me. Ribonuclease, which sorry sorry ও আচ্ছা সাউন্ড কম আসছে এখন ঠিক আছে হ্যালো নাও ইজ ইট ওকে okay ribonuclease as substrate of which is the which creates substrate of RNA. Okay, deoxyribonuclease substrate is, what is the substrate of deoxyribonuclease? Substrate of deoxyribonuclease?
DNA.
DNA. Yes. What is the substrate of phosphatase? Phosphate ester is the substrate of phosphatase. Okay. What is the substrate of glycosidase? Carbohydrates, glycosides, and polysaccharides. What is the substrate of collagenase? Say
Repeat.
proteins, not lipids. Okay. Collagenase enzymes produces proteins. Okay. The substrates there, lysosomes releases those. Okay. Then please next one, please. Next slide. Next slide, please, Ajim. So cytoskeleton. We have cytoskeleton as
The cytoplasmic cytoskeleton is a complex network of fine fibers that not only maintains the structure of the cell membrane, structure of the cell, but also permits into the change of the shape and move of the cells. The elements that constitute the cytoskeleton are microtubules, as I say, MMI, that means microtubules, microfilaments, and intermediate filaments. Previously, I said, and you need to learn the functions of cytoskeleton. Mainly, you can see the structures. You can see the structures. Can you see the can you see the microtubule here in the cell?
Yes, sir.
Yes, sir.
Yes,
Yes,
sir.
As it is
sir.
called, why it is called cytoskeleton? Because it is the skeleton of the cell. It maintains the shape of the cell. As our skeleton, body skeleton, maintains the shape of the body, it this cytoskeleton means the maintains the shape of the cell. That means site. Site means cell. Okay. So function of the cytoskeleton is maintains the structure of the cells, provides shape of the cells, permits the cell to change the shape, gives the support to the cells, and facilitates the movements of the organelles and the entire cell as well. This is the function of the cytoskeleton. Function of the microtubule individually, and function of the microfilaments and function of the intermediate filaments we also need to learn. Function of the microtubules is that they provide the tracks for transport the vesicles, organelles, and such as secretory granules, the mitochondria from the one part of the cell to another, and they also form the spindle which moves the chromosomes in mitosis. As we know, the cell division, about the cell division you have learned in the higher secondary levels about the cell division, isn't it? Didn't you?
Yes,
Yes,
sir.
sir.
You have gone through with the cell division and all
Yes,
those
sir.
spindle fibers. Remember, spindle fibers, and in mitosis we have 5 phases. Remember, what are
Yes,
those?
sir.
Remember the phases of my cell division, mitosis. What are those? Prophase, metaphase, prometaphase. Remember?
Yes, sir.
Yes.
Yes,
Okay.
sir.
Yes.
Telophase.
Prophase, yes, anaphase, telophase. Yes, the 5 phases. So, in case of mitosis, we got spindles. So the spindles is formed by these cytoskeletons. So microtubules can transport in both directions, and as part of the cytoskeleton, microtubules provide stability to the cells, and they assist in cellular movement by forming cilia, flagella, or centrioles. What are the centrioles? Do you know? Centrioles are the cylindrical structures composed of microtubules, and each centrioles consist of 9 sets of microtubules arranged in triplets. And functions of centrioles are mainly help in process of mitotic cell division. As you know, you have learned about the centrioles in the higher secondary levels when you have gone through with cell divisions play a role in formation of mitotic spindles of dividing cells, and they also have function in the formation of cilia, flagella, and certain types of cells, so almost similar to the microtubules. And functions of the microfilaments, do you know, participates in muscular contraction. It is the most important functions of microfilaments. Participates in muscular contraction. How muscles are contracts? By microfilaments. Okay. Can you see the microfilaments in the structure?
Yes, sir.
In the picture.
Yes, sir.
Yes,
So
sir.
microfilaments are the functions. They participate in muscular contraction. The muscular contraction occurs by these microfilaments. Due to the presence of microfilaments, many cells can perform such actions as endocytosis, in exocytosis, phagocytosis, movement of the flagella. Do you know what is endocytosis, exocytosis? Do you know? Do you know what is the exocytosis and endocytosis? Anyone?
No, sir.
No, sir.
Okay, when someone takes when some cells like cells captures the foreign body and it takes inside of it, and then it destroys the foreign body, and it releases after the destruction, and it purifies our body. This is called macrophage. I as I said in the previous class about the macrophage, didn't it? So the capturing and destroying this is called phagocytes. Phago means to eat, and sites means cells. The one cell sites eats the eats the foreign body or destroys the foreign body. This is called phagocytosis. And when it captures, it called endocytosis. When it expels out, it is called exocytosis. So endocytosis, exocytosis, all are the process of phagocytosis. So these are all those things are done. All those movements are done by the microfilaments in the cells, and movements of the flagella also done by the microfilaments. Flagella is the finger-like projections that helps the cells to float in the intercellular components or to transport substances in the intercellular components.
Yes, sir.
Okay, so that's all for the cells and cellular functions. You have intercellular connections. You don't need this. Please show the next slide, please. That's all for this. That's all for today. Yes, cytoskeleton elements you can see as intermediate filaments acts as a muscle contractor, and the intermediate filaments you can see microtubules, actin filaments. Actin filaments and are the most important thing in micro intermediate filaments. They help to contract the muscles. We'll discuss it in details when you will go through the mechanisms of muscle contraction and relaxation in physiology. And in the next class, we'll go through the transport of substances through the cell membrane. We'll go through the transcytosis, and we'll go through all those things, phagocytosis and others in details, inshallah. And we'll go to the